Here we are on this practical. Your job is to carefully watch and ask your questions and see how these questions will be handled. Just see you in the comment section as we proceed. SirMajesty.Sure Science channel is here for you, and we are transmitting live. So your job is to get your questions. Watch now. I will give you the details of these questions with explicit explanations, of course. So over here, this is the paper tree. Remember, it is 80 marks, 80 good marks. So your job is to listen well, ask your questions where necessary. Then the summary of the answers, I will use board now to explain them. And then the summary of the answers, of course, comes. Okay, here we go. Okay, so we now need board. I will be reading the questions, solving them gradually. If you ask any question, you pop up in the comment sections, of course. All right, here we go. So these are board. I'll bring in the questions to read from. All right. Okay. So the first questions there, remember the list of specimen. We have the specimen A, because the list of the specimen will help us understand the answers. Just to remind us again, to help us to get the message. Okay. So we have the first question that says that you should study specimens A, B, and C and answer questions 1A to 1F. They tell us, observe specimen A carefully with the aid of magnifying lens and identify the specimen with a reason. We are required to identify here with a reason. Then they say, what is the mode of nutrition of specimen A? Remember that specimen A is being weevil. So if you are online, you can share this live stream to help others in this class. We are giving you the best service. Just stay tuned and listen to their answers. Here, specimen A is being weevil. Listen to the answers correctly. And the summary of the answers to this will be posted on my separate channel. This channel is science channel. SirMajesty.Sure Science channel. It's meant to handle sciences, but because most times we are not known for exam, I created a separate channel where I'll be doing next section. We'll be handling every answers there. Here we'll be focused on dealing with sciences. So the channel where you get the answers, the summary of the answers to these questions you are seeing, is you go to, like, I will answer everything here, but the summary where you can get the written down, the compiled answers to the whole questions, you visit Majestic Tutorials. Just Majestic Tutorials. You search for it, or you rather, I'll keep the link immediately after this live stream. It will be available. Then for the drawings, I'll draw most of the specimen here. The drawings, I've made the soft copy drawing, which is still available at Majestic Tutorial. This tutorial is still owned by mine. The main aim is to do with theoretical work, exams, teachings are there, but this channel you are watching now is going to handle only practicals, science channel. So that's the name, and it's going to focus just in dealing with what sciences. Okay, so the first one says that we should study specimen A with the help of magnifying lens, and specimen A is being weevil. They now tell us to do what? To first one is to identify with reason. First of all, specimen A is a being weevil. Reason is because you take other features of uh, beetles and weevils. They belong to the coleoptera. At least it has small packed body, then it has a what we call a elytra. So specimen B is a being weevil. It has a hard dark oval body characteristics, which is the characteristics of the members of the coleoptera. So that's the answer to that question. The other one said, what is the, looking at the other question, he said, what is the mode of feeding of specimen A? Specimen A feeds on grains, so we can call it a granivorous somehow. It feeds mostly on grains. Then I say feeding on grains. That's what you will answer there. That's the mode of nutrition. Though it is heterotrophic, you can also say it is a heterotrophic. It depends on already made food. It cannot produce its own food, but it largely feeds on what? Grains. All right. So the next thing there is what is they say highlight any two adaptive features of specimen A. Highlight any two features of adaptive features of specimen A. What are the adaptive features? Hard exoskeleton protects it against physical damage. Well-developed mandible used for boring into seeds. Well-developed mandibles used for boring into seeds. Then strong legs with spines, then which aids in a movement and anchoring to the seed. So these are the structural adaptations that helps the survival of the being weevil. Remember, it belongs to the largest order. You know, in the insects, the largest order in insect family, the largest order in insect class is Coleoptera, while the largest class in the arthropoda is actually the Insecta, while the largest phylum among the whole animal phyla is actually Arthropoda. Okay, so the next question says, what is the habitat what is the habitat of specimen A? Habitat of specimen A, I warned earlier, don't be too broad. Don't tell them terrestrial. No. Rather, you say, where's the habitat? Stored grain, stored legumes, stored beans, legumes, and other things. No, it's a storage pest. So it lives inside stored grains or stored legumes, stored cereals, or stored beans. That's the habitat. Identify the phylum and class of specimen A. The phylum of specimen A is automatically Chordata, and then the class is Insecta. Okay, so I'll be reading the questions and handling them gradually. So we have the other one for food test. Looking at the question towards the end, we have food test, which instructs us to do a hold specimen G vertically and gently break the shell at the smaller top portion of specimen G. Withdraw 2 cm cube of the transparent liquid using a syringe. Transfer it to a small beaker and dilute it with twice the volume of water. Add sodium hydroxide to the resulting solution, followed by 1% copper 2 tetraounces of a drop by drop while shaking vigorously. Then the same question says to II, say to 2 cm cube of specimen H, add 2 cm cube of sodium hydroxide solution followed by 1% copper 2 tetraounces of a that's copper sulfate drop by drop while shaking it vigorously. So in this case, there is a kind of, there will be control experiments and there will be the main experiment. So this is Burette test. Specimen G is the egg, of course, and definitely contains protein, while specimen H is water and is going to show absence of what? Protein. So for specimen H, question may not likely come there. Please, this is meant to give you the final revision that you need for the exam. Okay, so your job, remember where to visit. Just immediately I drop this live stream, I'm sending the compiled answers to these questions you are seeing. So They say record your observations and inference in the table below. Remember you have to have your test, observation, and what? Inference. As usual, there will be. So you are on with SirMajesty Easy World, of course. But you can visit my new channel, Majestic Tutorial, to get this. So we have the table for test, observation, and inference. So remember, the other one is just a specimen G. Specimen G, that's a plus sodium hydroxide plus 1% CuSO4 plus a shake. You say what? Purple or violet coloration. Purple or violet coloration. And this violet coloration automatically confirms what? That what is present is you write what? Protein present, because this detects at least presence of 2 polypeptides. All right. So then the other one, which is specimen H, is water. Okay. All right. Okay. All right. Welcome back. Welcome back. Welcome back. So remember to welcome back. Remember to read the questions very well in the question paper before providing their answers. That is what you are advised to do, of course. So if you don't read questions well, you might not get their answers correct. Okay. I hope that what I'm writing is not actually on the reverse. Maybe I confirm it here. If you have any question or challenge, you can bring it in. I will see your questions. Okay. I wish you the best of luck in your examination. Okay, it is it's still showing, the screen is showing on the reverse. That means we can't read it. I need to twist the camera the other way around so that we can see the correct thing there, because what I'm writing is showing on the opposite. All right. Okay, I think this will be very fine, so you can see what I'm writing. All right, we are back. So please, I'm setting this thing to serve you well, to give you the best service. All right, I think we got it. So it becomes too obvious. Now, the question is all about Burette test on protein. So specimen H is plus sodium hydroxide plus 1%. Remember to read the questions well in the question paper. This class is meant to prepare you at least so that you don't fail. 1% Cu, just the same thing you did there, plus shake. You still say what? What will happen? Blue coloration remained. The blue from the copper 2 will remain because copper 2 is blue. So protein what? absent. So that's what you do there. Now, other likely questions that we will handle very well. Then we have handled they say the economic importance of specimen A. Okay, we say store food product, particularly dried legumes and grains. Okay, the habitat of specimen A. We have finished that one. I told you not to be large. Just say it is found in stored foods, particularly dried legumes and grains. Then what are the economic importance of specimen A? The economic importance is that it causes significant loss in stored grains by boring into seeds, reduce the quality and market value of infested grain. Then the question C, I say mode of feeding in specimen A. I've said that it is actually granivorous. Granivorous, it is feeding heavily on dry seeds, more of the grains. Okay. Then what are the effective methods of controlling specimen A? The effective methods. All right. The effective methods for controlling specimen A, at least storing in an airtight what? Container, fumigation of infested grain using insecticides. So this one is another one. Then you can use neem leaves to scare away the insects before it could enter. If you tightly store it in a container, it won't have any entrance. Then the next question says the phylum and class of specimen. Okay, we have finished all these things. They might ask us what's the subkingdom, if at all. The subkingdom should be invertebrata. Some might tell you it is metazoa. Then they might ask us to draw and label specimen A. If you draw specimen A, use magnification of about times 20 or times 15. So for specimen A, the drawing, remember to obey the size of the diagram. Some of you will ask, is the title part of the size of diagram? No, title is not part of it. Maybe they tell you to draw 6 to 8. I want to draw here. Maybe this is my 6 to 8. Title is not included. Magnification is not included. It's only that diagram that we are going to do what? Maintain that size. So title is outside. So the title will now be what? Diagram of, in capital letter, diagram of a specimen A. Underline better. Therefore, don't exceed the mark of that height. So which means I'm going to have something like the head. Then we have the thorax. Which gives us this V-shaped. Then on the head, we will now have the antenna. Remember to draw antenna in just double lines. It's a must. Don't allow your drawing to exceed the mark. Maybe this is my mark. I have to minimize the proportional. Over here, you have to introduce the compound I comes in here. Then the jointed legs. The first one faces up. Then the next, try to be proportional. Yeah, the much you can. You now write mg, can use times 20 or times 15 in this case. Label carefully, remember no shading. This is antenna. You must rule this your guidelines. Your guidelines must be ruled. Okay, your guidelines must be ruled. Antenna, be careful with your spelling. Antenna. Then here is the compound I. Remember to get the compiled answers to these questions, you visit Majestic Tutorials. I'll give a link. I'll leave a link immediately after this live stream, or you can search Majestic Tutorial channel. Please, I created that channel to do with only everything theoretical. This channel is actually science channel, so it's focused on doing sciences. If you check this channel, you see most experiments. So here is the head. Remember to avoid cross line of labeling. Here is the thorax. Your line of label must touch what is labeling. It must not hang. Thorax. Here is the abdomen. Remember, any two correct labeling equal to 1 mark. In biology, labels are marked half-half. Any 8 correct labeling. Most drawing don't exceed 8 correct labeling. Then this wing is called the elytra. So here is the jointed legs. If you draw one, you just make it jointed leg. Okay, so you maintain the size. That is it for specimen A. We go to the next likely questions. The summarized answers I told you is already there for you when you visit that channel. We look at identify specimen B with reason. Specimen B is a seed with holes. Reason: the presence of tiny round holes indicates infestation by weevil, because being specimen B is just a bean seed with holes. So what made you think that is infested? They might ask you what condition is in B. You say the condition is infestation by weevil, of course. Give reason for answer. The presence of hole indicates that what is infested by what? Weevil, or that is infested by storage what? Pest. Then I say state 2 effective methods of controlling the condition shown in specimen B. Here you say use airtight. This is similar to the other question we have answered. We need to use airtight storage containers to prevent infestation. Apply natural insect repellers such as neem oil to protect stored grains. Now, the next one says state the economic implication of the condition in specimen B to farmers. The implication is that it leads to what? Loss of seed viability, leading to reduced crop yield. Like the viability of the seed might be lost. Remember, infested bean can germinate, but it is not sure. The germinating chances reduces, so it is causing low or reduced crop yield. So we have also decreased market value of the infested seed. So when you take this bean to market to sell, you know what it means. Nobody would like to buy such bean that is infested by weevil. If you must sell it, you sell it at a very cheap price. Okay. Now the next one says if the dormancy of, which means the economic importance is that it affects the farmer's income, it reduces the market value of the bean itself. So if the dormancy period of specimen B is 2 weeks, state what will happen after 2 weeks of sowing specimen B in the presence of unnecessary conditions. Give reason for your answer. This depends. You can now say if the seed, if specimen B is still viable, because the infestation might damage if it affects the radicle and plumo, it damages it. Okay, so if specimen B is still viable, it will germinate. Now, the next question, but if it is not viable, it will not germinate. Why? Because only viable seeds can germinate. Is it not only seed that is alive that can germinate? Yes. Then the next one says the seeds will likely fail to germinate because of the infestation or damages to the embryo. You can provide such answer also. Then the next question says if B is still viable, Mention other factors necessary for germination to occur in B. Remember, this specimen, they might leave the specimen and start asking you what connected the specimen. Like you might see further, they will start asking you questions on birds in general. It's possible. So a link. So when you see any specimen, you study everything around that specimen to do well. If B is still viable, mention other factors necessary for germination to occur. They are asking you those factors necessary. Adequate moisture, oxygen availability, and then suitable temperature. Remember that sunlight has no effect on germination. Without sunlight, seeds germinate. That's why seed can stay inside your room, in the dark kitchen, in dark corners, and germinate as far as these conditions are there. But plants now need sunlight to maintain growth when they start to photosynthesize. Then the next question says, identify whether specimen C is a dicotyledonous plant or monocotyledonous plant. Give 3 observable structural reasons for your answer. Specimen C is water leaf what? Water leaf plant. So is water leaf plant a dicot or a monocot? The correct answer is that specimen C is what? Specimen C is a dicotyledonous plant. The reasons are, what are the structural reasons? Okay, the presence of I'll draw the specimen C. The presence of broad leaves with reticulate venation, meaning network venation, then taproot system, then presence of vascular bundles arranged in ring within the stem. This is not observable. Don't say this last one. Like presence of a vascular bundle arranged in ring within the stem. You are not going to say this unless if you are told to cut, which is going to be part of the question, to cut the stem transversely and observe. That's when you now say presence of vascular bundle arranged in a ring within the what? Stem. Okay. They now say what type of seed plant is specimen C? What type of seed plant? Seed plant. This is where we get it wrong. Seed plant. When we use the word seed plant, some people will straight away just say it's monocot. Monocot is not an answer here. Seed means sperm. Plant means fighter. So what do we mean? Spermatophyte. Spermatophyte means seed plants. So spermatophytes are the seed producing plants. Spermatophytes are the seed producing plants. Permit me to clean this. Spermatophytes are the seed producing plants. So when they tell you the type of seed plant that is there, you now say we have 2 types of seed plants. Seed plants are divided into 2. That's those plants that produce seed. Those that produce seeds covered in fruit wall and those that produce naked seed. And those of them that produce seed covered in fruit wall are called angiosperms. So, and the other one is gymnosperm doesn't concern us here. So the answer to that question, the type of seed plant is what? Angiosperm. Give reason for your answer. Presence of flower. This angiosperm means flowering plant. Presence of a flower makes your answer very, very correct. Okay, so seed plants, don't say monocot or dicot. All right, so I said angiosperm as flowering plants, but it means those plants that produces seeds that are covered in fruit wall. But the other one, gymnosperm, they produce seeds, but they are naked seeds. They are not found inside the what? They are not found anywhere inside the there is no fruit. They produce it, but they don't have what? Any fruit. Okay. For the drawing of specimen C, you might likely expect that the whole plant. And remember, specimen C is water leaf. They can compare it with orange plant itself. So, or compare it with a pineapple plant itself, because pineapple should be a monocot, at least. Looking at the features, the leaves are spindle in shape. Okay, but this is a dicot. So drawing this, we might have just maintain the basic things that are needed. Remember to watch my video on the rules guiding biological drawings. It is tagged, don't lose 35 marks, because drawings, you'll be drawing 2 specimens. Out of all these specimens provided, you'll be drawing 2. And these 2 will carry 22 marks. So out of the 80 marks, drawing will give you just 22 marks. So why are you going to fail again? Grab the smooth 22 marks. That's why I'm here. So we have to put our normal venation, come down, the same thing applicable here. There should be this stalk. Please show the flower, the leaf stalk. Remember, you are not expected to draw like an artist. Your job is just to make those basic features that are expected to be there present. And then there are these roots. Then endeavor to show the taproot system. So in your labeling, you remember this one is easy. If they ask you to draw, you just label tap root. What's your magnification, please? Mg here you can use times half or times 0.2 times half. Then here, please just put the venations in each case for the leaf. Remember, no shading. You have to write diagram of, please. So tap roots, while here is the stem. Then here is the axis. This angle is axis. But if you draw anything here, you can say auxiliary board. Maybe if you indicate something sprouting out to form a branch, you can call this auxiliary board. But this angle is axis. Then this base here is called node. Then this space here is called internode. So these are the basic things that you are required to elaborate. Then if you like, you can now put flowers. Just tiny structure. Don't shade, please. You can label flower. Okay. So that's it for specimen C. Okay. Let's continue. Then the next question is we are required to state what type of seed plant is specimen C. I've answered you that. Give one observable reason for your answer. The correct answer is angiosperm. Reason, it has flowers and seeds enclosed in fruits. Okay. Though we are not observing those seeds there. State two adaptive features of specimen C to its habitat. Number one, broad leaves for efficient photosynthesis. Number two, soft succulent stem to store water. As we know, water leaf we used to cook and prepare rice and other delicacies over here. Soft succulent stem to store water. Then C, we can now see question that say identify the phylum, class, and family of specimen C. The phylum is Angiospermophyta, or just Angiospermae, which means flowering plants. Then the class is Dicotyledonae, meaning it has two seed leaves. So, okay. Then they might tell us to cut the stem of specimen C transversely. Observe with a magnifying lens. Draw 6 to 8 cm diagram of the cut what section. Okay. Then the next one, this one is you are expected to cut this, though this is unlikely, but we prepare against all odds. To use a scraper, you cut this and observe. Your diagram should just show the ring arrangement of the die cut stem. So maybe I will minimize this. You have to show them that the vascular bundles are arranged in a ring form. Remember not to put anything like root hairs, because we are talking about stem. There is nothing like hairs or root hairs there. Alright? So you show your vascular cambium. Remember, no shading. So any cut section, any cut specimen must be in double line. So these are the rules. There will be white pitch. We can now label basically, but these are the features of die cut word stem. Okay. Make a label drawing 8 cm of a complete specimen C, which I've drawn there. Detach a single leaf from specimen C and draw a labeled diagram of detached leaf. They might tell us to remove one leaf here and draw. That means structure of a leaf. So, but you should be careful not to confuse leaf with feather. Or some people, when they want to draw feather, they will draw it like leaf. Meaning remove one leaf here. This is unlikely and draw. So while drawing a leaf, maintain what? The apex. This is what you are required to know. The midrib, you show it. And then the leaf what? Stalk. You might show where it's connected to. Then you show your reticulate venation on network what? Venation. So these things are not as complex as it may appear. So they are very simple. Here is the apex of the leaf. Then here is the leaf blade. Here is our midrib. So just watch, go to Majestic tutorials, you will see the drawing of all the specimens. So to save time, I will move on. But for any specimen that is difficult, if you ask me to draw any, I'll help you out. There are cheats for drawing all of them. Thank you for being there. I really appreciate you. The best of luck is what I have for you. You're going to do well in this exam. Remember, biology is 80 marks. Don't joke with it. Then I'll say state any two methods of propagating specimen C. How can we propagate water leaf plant? Stem cutting and seed propagation. You can use the stem. You can cut the stem to bring new plants, or you can use the seed. Okay. Mention any two, which means in specimen in that specimen C, we can have vegetative propagation there, which is that stem cutting. Then mention any two perennating organs. Perennating organs in specimen C, meaning organs that can survive one season to the next, one planting season to the next season, that will help organism survive within 2 years or 3 years. Remember, we have biennial, perennial, annual. So any plants that can live from one season to another, what are those things that will remain? You know, the leaves will not be there. So the correct answer, the perennating, perennating. The perennating organ there is the what? The roots. Okay. So the roots and stems. Then the next question says, identify specimen D, providing a reason for your identification. Specimen D is the queer feather of a domestic fowl. Reason, of course, you can say many things there, but because they said queer feather, just any feather that have hollow shaft, presence of hollow shaft. If you press the shaft, you can see hole. Initially, it was commonly applied in writing as pen, that's in the ancient time. Okay, so you can also say it has a long, stiff shaft with barbs arranged in what? On both sides. Now, the other question says, state the phylum and class of the organism that possesses specimen D. Imagine now, no, specimen D is now queer feather, and we know that the only organism that possesses feather, as far as biology is concerned, is bird. All birds, if you see feather, that is a bird. Just like if you see hair, that creature must be a mammal. But if you see bird, there must be what? A feather. Even when it's not using the feather, there might be scanty feather. So they might use this opportunity to question us on birds. State the phylum and class of the organism that possesses specimen D. Phylum, Chordata, reason because it has a backbone. Then class, what's the class of bird? All the birds belong to one class called the aves. The aves, okay? Detach the leaf from specimen C and compare it with specimen D. State one difference and one similarity between the two. What are the differences between feather and then that of the leaf from a water leaf plant? Okay. The similarity is that both have a central shaft or rachis. in feather and midrib in the leaf. So both have central support. In leaf we call it midrib. In the feather we call it the shaft. Okay. Then specimen D has barbs, while the leaves has a broad flat lamina. What we call lamina is the leaf blade. Leaf blade is the lamina. Okay. So next one says state one function of specimen D to the organism that possesses it. What does feather do to the organism that possesses it? That is the functions of feather to the birds. Your job is to start stating all the functions of feather, you know. You now say it provides what? Insulation and aids in flight. It helps birds in flight. It provides insulation. It helps to maintain the streamlined body of they say state one function of specimen D to the organism that possesses it. Please don't use it helps. So just say it provides what? Insulation. Do you understand? It provides insulation. Remember not to use it helps. I made the mistake just now. Why marking Waec? When they tell you what's the function of this structure, don't say it helps, it aids, or it facilitates. Just say it is. So please, the function of feather to bird is that it provides insulation. That's a good thing. It is used for flight. When you see it at the wings or at the tails, then you can now say it is used to maintain, yes, the streamlined shape of birds. It is used for beautification. Okay, these are the general functions of feathers to birds. Now state one structural modification of specimen D that adapts it to its function. All these functions we mentioned, what are the structural modifications, which means state the parts and say what it does. You now say the answer is presence of interlocking barbs that form a smooth, continuous surface for flight. Okay. Those your questions, I'm seeing them here. I'm seeing them live. That I will forward it. I told you to visit Majestic Tutorials. Somebody said do for chemistry too. I will. But disclaimer, whatever you saw before clicking this video is just to help you join the class. You will not regret joining this class. This class is actually what? This is a mainly general revision. Okay? Not actually the real questions. I'm sorry if I made you think it's the real question, but of course it's about 90 something percent sure. It's just to help you learn. Okay, so please, I will do the same. You know, this is Her Majesty. Even if I have the real question, I wouldn't do that. I'm not involved in a examination malpractice. So, but I teach you, I do experiments. I'm not saying I'm holy. I'm doing the much I can to help education. So I can't kill the same thing I'm helping. So please show us the questions for more clarity. Yeah, you visit Majestic Tutorials. That's where you see the questions. The other person say, will you do alternative to practical next? Okay, alternative to which practical? If you chat me, I'll send you, not just say alternative to practical, I'll send you the video link for so that you know how to do alternative to practical questions. There is nothing there, and somebody cannot do otherwise. You cannot predict what is there, but we know the range of what they will ask. Okay, I see all of you. Hello, sir. Thank you. I'm saying hello to you all. So please, if you send in chats with time to time, I'll visit to see what's going on there. Okay, so we go back to continue. They say state one structural modification of specimen D that adapts it to its function. Presence of interlocking barbs that form a smooth continuous surface for flight. Please, I will send these questions direct to that Majestic tutorial. If you visit that tutorial, I created that channel newly. Please subscribe to that channel. Subscribe to this channel also if this particular live stream have helped you in any way. Please subscribe and share to help others also. But if you visit that channel, I'm begging you, please just subscribe. My aim is to do sciences here, experiment, research, amazing entertainment with science projects, but anything theoretical class, tutorials, teaching, exams like Waec time, we will be doing it there at that Majestic tutorial because I created this channel for science. But due to our people don't have this positive attitude towards science. When I do scientific things, I might spend money, do researches, and upload a wonderful video. You can see the foreigners only watching, and people don't care about that. But you see them complaining they don't do practical for us in school. But this channel has been there for you. Go through this channel. You are watching this just because of exam tomorrow. But I have what it takes to make you scientists. Certificates cannot save us. Let's join hands together. We are far behind. Africa is far behind. We should focus on science more than we focus on certificates. I don't know why we so much love this. And this is why people engage in malpractice and do all sorts of things, because we love these certificates. We don't even love the science. Please, it's high time. Support this channel. This is Sir World. I had many scientific experiments, many things I've done here that I needed you people to promote. It's only the external that promote, but I can't jump you people. It is you people that will promote me before the whole world will see me. So thank you. I'm just pleading you, if you are a lover of science, stay close. But anything tutorials, I started teaching tutorials here, bringing in exam things like this, because our people have negative attitude to learning. But even if I finish this now, I will upload one wonderful scientific video very shortly. You see that there will be low turn up in watching it. So this is science channel and not just tutorial. So that's why I created the other one separately to serve you very well. Majestic Tutorial, Majestic. I'll send the video link, the channel link, down here in this video just after it has been fully uploaded. Okay, remember we are discussing the structural modification of the feather that adapts it to its function, presence of interlocking barbs that forms smooth continuous surface for flight. Make a labeled diagram 6 to 8 cm of specimen D, that is feather. So it's part of what you get at the Majestic tutorial is already there, the drawing and the labeling. Then the next question say, what type of fruit is specimen E? This time around, you can answer many things there. You can say succulent fruit. That specimen E, remember, is pineapple, is a succulent fruit or fleshy fruit. Okay? Reasoning, because the pericarp is juicy. Pericarp means the total fruit or the pulp is juicy. Or you can say that pineapple is a multiple fruit. Reason, it develops from what? The fusion of multiple flowers. It developed from inflorescence into a single fleshy fruit. It was formed by many fruits. That's why if you go there, you see that my diagram on the longitudinal section of pineapple is longitudinal section of pineapple. Please remember, if you are told to identify that specimen E, don't just say pineapple. Say E is the longitudinal section of pineapple. You must put that longitudinal section, please. Okay. So you can also tell us that pineapple is a multiple fruit. Reason, it developed from what? The fusion of many flowers. Cluster of flowers is called inflorescence and formed one single fruit. Just like soursop, your breadfruit, these are all multiple fruits. If you don't call them multiple fruits, you can call them composite fruits. State any three differences between E and F. That is, differences between pineapple and orange. Remember, the pineapple is a longitudinal section of the specimen, while the orange is a transverse section. Number one, specimen E, pineapple, is a multiple fruit, while specimen F, orange, is a simple fruit. It is a simple fruit formed from a single flower with syncarpous ovary. Syncarpous ovary, that is many ovary fused together. Then pineapple develops from a cluster of flower, while orange develops from a single flower. So they say the differences, we are going to use observable differences, please. What I said here are not really observable. If the question say observable, you say presence of crown in pineapple, absence of crown in orange. You also say presence of bracts. When you watch that video, you see the drawing. Presence of bracts in pineapple, absence of bracts in orange. Okay, so these are the common one. Then presence of hair-like scales or scaly structure in pineapple, absent in what? Orange. Presence of rind in orange. There is what we call rind. ND. Rind. Rind means mesocarp and epicarp fused together. So rind is present in orange, that is, but is absent in what? Pineapple. Rind. That's why peeling orange, you can never see the green part without seeing the white part. The greenish outer part is the epicarp, then the white soft part is the mesocarp before you go to the juicy endocarp. Okay. Mention two similarities between specimen E and F. Both contain seed, both have fleshy edible what? Parts. They have fleshy edible parts. Both are fleshy fruit. Both are juicy. Both contains sources of vitamin, okay? State any three structural adaptations of specimen E. One, specimen E here, you say thick spiny skin to deter herbivores. Thick spiny skin, that means also we can say spines or scale present on E, which is pineapple, but absent on F, which is orange. Then the other one says fleshy pulp to attract seed dispersers. So it is edible. E is edible to attract seed dispersers. Okay. Then fibrous core for support and nutrient transport. Okay. Then make a label drawing of specimen E. That is the longitudinal section. It is also part of the drawing you find there. Now we say identify the phylum, class, and family of specimen E. Of course, it's similar with orange. E and F, that's orange, even water leaf, they are all angiosperms. So we have three angiosperms, specimen C, specimen E, and specimen F. All the seeds provided or all the plants are all flowering what? Plants. Then they said the next question said placentation. We are going to see the type. So placentation type in specimen F, axile placentation. The same thing is observed in specimen E. Both E and F all have what some authority might say is parietal placentation, but it should be axial placentation. Though the other one is a longitudinal view, the other one is a transverse view. Okay. Now, what are the observable similarities between E and F? Both are fleshy and edible. Both contain seeds within their pulp, pulp, that's within their succulent parts. Then, the classification of F from kingdom to phylum, this one might take a longer time. Just know that you are required to know the scientific name of all these citrus species. Just remember SPP can serve citrus species because they just say orange. They did not specify. So citrus SPP is the scientific name. Why the mango should be Onanas comosus. So the names are included in the guide. What are the economic importance of specimen F? Source of vitamin C, of course, for human nutrition. So the juice is the juice are actually that specimen F is used for production of juice. We know that we drink juice where pineapple is part of it. If you hear about Five Alive, many brands that uses pineapple, so can be used in juice production. Then it is source of vitamin C for human nutrition. That is the economic importance of F. Remember, economic importance includes both advantages and disadvantages. Then types of fruit in specimen F. Specimen F is orange. It's a berry, but you need to be more specific. Don't be too wide. Orange is Hesperidum. Hesperidum. Why is it a Hesperidum? Because it has what we call leathery ring. Orange is nothing but what? Hesperidum, because of the presence of leathery what? Ring. Okay. Let me check the comment section. Oh, wow, a lot of comments. A vessel for divine love. I'm illuminated by the radiance. Oh my God. Oh, Mama Ahims, you're overpraising me. Thank you, thank you. Hello, sir. With due respect, why did you start your class by writing? I'm so sorry about that. I never knew. I later saw it here, and I have to change it. Okay, don't mind me. I was just saying since my face is not on the lateral inversion, everything's okay. So pardon me about that. So good evening, Loretta. Good evening too. Wow, goodness. Is happy to see this. Okay, on time. But remember, it's a guide, though. Please read questions well in the question paper. Whatever you see before clicking the video is just to help you learn. Okay. Then share it. I'm sure you come back here with testimony. Okay, these are the ones we have freshly. Let me go back to our classes as we are almost concluding. The other one says the type of fruit that specimen F is, that is orange, Hesperidum. Reason, it has leathery rind. Leathery rind. Like the rind, if you peel off orange, when we are still at a younger level, younger than I am now, we don't use knife to cut orange. We peel off, we remove the rind and shear the orange into chambers. Imagine, we are doing biology unknowingly. That time we will share it, because if you peel off orange, I don't know if you have been so rugged to experience that. Rained entirely, you have the what exposed, the endocap exposed, but the endocap have what? Lines. So we never knew that we now start detaching it. Do you understand that each one you remove is called a carpel or a chamber, which was formed from a single flower? And if you go to that flower, it will have the number of stigma, which is equivalent to how many chambers. So by then, you just come out one chamber or one carpel and give to your friend. So this shows that orange have a syncarpous ovary, that is many chambers fused together. Science is so lovely, but we never knew. Okay, let's continue. We say it has that leathery rind is what you remove before you start dividing it into that chamber. Okay, so leathery rind, that's the reason we call it Hesperidum. We have another one that doesn't have leathery rind, like the purple. Stay tuned to watch my detailed video, or practically when I will handle fruits. But problem is that we don't love science, we just love certificates and exam. Here I'm going to, when I will handle fruits, I will use with projector everything, fruits and flower. You see the sciences behind it. Remember, I take you on not only biology but in chemistry, of course. Then the next question say, what's the phylum of specimen C, E, and F? I've said it already. They are all angiosperm flowering plants. Specimen E, pineapple, they are all the same group. Now, reason for all of them being angiosperm is that they are all flowering plants. What are the structural similarities between specimen G and J? G and J. G is just a raw egg. Remember, just say egg. You don't have what it takes to say raw until you are told to crack it. You can now see which is what I started the class with. Okay, so structural similarity between specimen G and J. One is that both contain an albumen layer. This is when they tell you not to observe it, but please, if they tell you what they are observable similarity, just say presence of shell in both, because you can see shell here and you can see shell in the other one. Don't say albumen, because you can't see in that G unless you crack it. Okay, then both are edible, of course. Both are still used in baking as culinary and making a cake. I don't know how to see it. So what are the observable differences between specimen G and J? Okay, you should know that G is a longitudinal G is a whole what? A whole egg intact, while J is egg cut on the longitudinal section. Now, the next is the Burette test, which I've done already. The Burette test is used to detect protein because it's sensitive to at least two polypeptide bonds. When it's present, that is when this one works, the Burette test. Okay. With this, we also say, what are the economic importance of specimen G or J? That is, it is source of protein in human diet, is used in baking and culinary applications. Of course, you use it for baking, and it is also source of protein. Then it's also source of income to what? Farmers. These are the things we need to know. I am trying to draw the curtain here. You might be told to draw the longitudinal section of that specimen J. The drawing of all of them are there. Okay, there is also a question that said that we should compare between what state two adaptive features of specimen C to each. Okay, we have answered this one. Please be practical in mind. I want to remind us again, you are going to be handling practical biology, which sums 80%. Theory is 70, and the objective is 50. This is unlike chemistry and physics. Physics and chemistry, our practicals in chemistry is just 50 marks. Theory is 100. So don't joke with this practical biology. I want you to get A. Then when you come to this practical, it's made up of section A and section B. The section A is 50 marks. Two questions, 25 marks each. Then the whole exam, two drawings out of the whole specimen provided for you. Two drawings, which will score you at least 22 marks. One drawing is usually 12 marks. The one you will label about 8 parts. Then the other drawing will score you about 10 marks, where you label about 4 parts. Okay, so I can't start the whole entire video on the way you present your answers. It's not just knowing the answers, but the pattern of presentation of your what answers. So before I leave the live stream, let me see if there is any other new comments. Okay. Okay, thank you guys, thank you guys. Say explain specimen B well, sir. Specimen B is I explained it well, of course. Specimen B is just a bean with hole. It means a bean seed that is infested by weevil. What is your reason for saying it's infested? Because of the presence of the holes. Then they might ask you, how do we control the condition there? By using insecticide, fumigating it with insecticide, and storing in an airtight container, or treating with neem leaves to scare away the insects. Okay. So the other person said, please explain space. Okay. Well done, sir. Can I really see the questions, sir? Thank you. I told you where to go to see the questions. Majestic Tutorials. Shortly after this, I'll share the link. You just search it, Majestic Tutorial. You see my face there, of course. The drawings and most of these questions I said here are already there, but some are not there yet. But I'll still upload more there. I'll still do the same thing I did here with chemistry. So please, your job is to go to that my new channel and subscribe. And I'm encouraging us to be in love with science more than we are in love with certificate. I always say, I have the certificate, my father have it, my mom have it. The same is applicable to you. You are now struggling to go and get your own. Fine, these things are okay, but they won't save our nation. As you can see in the world today, the world is driven by technology and science. But we are still busy establishing religion. Okay? What is uncalled for? If we must change our nation, we must focus on science. I'm doing this just to lure you because this is the only condition you can hear me out. Do you know there are wonderful videos here on this channel? If I start mentioning them, there are many of them where you can generate electricity from ordinary chemical reaction. I can't recall many of them, but there are experiments that you can look at and say, wow, can equally learn how to produce soaps. They are all here. To know the differences, practical classes, there are many of them here because this is Science Channel. Please support me, grow, love science. Let's help Africa. Is this thing happening to us? Are we aware of the situation of this? But I gotta stop the live stream before I say one thing that might implicate me. But all the same, please subscribe and share this video. Best of luck in your exams. Remain majestic. Bye for now.